All right. Hi, I'm Snigtha, and I'm one of the co-founders of YouthSTEM. And I recently took AP Biology and found one of the labs that we did very cool, very simple. It was definitely great as a learning tool for the topics that we were covering within that class. So if you're interested in biology or just wanting to do a fun lab just to get the hang of doing these labs and experiments on your own, this is a very simple project for you to do, and it's definitely loads of fun. And so, like the title says, as you can see, it was about modeling osmosis using eggs. There are definitely other materials that you can use that aren't eggs. In the first iteration of this experiment, actually, my team, we used apples, but we realized that it didn't have a very permeable membrane, which is essential for this certain experiment, since osmosis needs to occur through a membrane. And definitely that's the term that you should familiarize yourself with a little bit more before doing this experiment specifically. But it's a pretty basic biology term, so if you're taking biology, you probably already covered it already, or something you'll learn in the near future. But yeah, we used eggs because their membrane was basically the easiest to work with. Um, and yeah, eggs are very cheap, making them great to be using. So to go on to the next slide. Yeah, so this is just an experiment overview. Like I said, we started with apples, which is shown in that top photo, but then we redesigned it using eggs. And you can see that we had 6 different eggs here. And the reason, again, why we chose these eggs is because they have a large initial mass, meaning that there can be significant changes that are given to them. When we used the apples, the pieces were too small, which just it didn't work out. We didn't get noticeable results. These eggs were also able to just be soaking long enough within the samples, and we'll get into what the experiment was, so that'll make more sense as we go on. And of course, they have a semi-permeable membrane, which is essential for osmosis. And so the independent variable and the dependent variables, I'll go through that. If you are new to experiments, these are some of the first things that you need to define. So basically, what are you looking to do? What are you looking to accomplish? In this case, it was to model osmosis and see how that can just function using real materials such as eggs. And then also identify what your independent variable is. So what are you specifically changing? What are you controlling and changing for this experiment? And then your dependent variable, which is going to be affected by how you control that independent variable. So our independent variable was the concentration of the solutes, and the solutes we were using in this were salt and sugar. And then our dependent variable were the changes in masses of the eggs, since we were measuring osmosis and seeing how much water was being transferred into the egg or transferred out of the egg. So the basic overview, the next part of this project, or any project, is to have a basic procedure so that you understand what you're trying to do. So this is a very basic procedure. Definitely, if we were to write a full lab report about this, we'd want more exact measurements, more exact descriptions on the handlings of the equipment, just to make sure that it is a step-by-step procedure that is easy for anyone else to follow. But just to simplify it down so that you can take your own spin to this experiment and just use this as a basic guide, we just masked all of the 5 eggs so that we knew what they initially started with, and we placed each of them in their beaker for 1 hour. We'll go into those beakers after. And then we took out the eggs and masked them again to see how much they changed. Basically, by measuring the masses, we could see if a lot of water went through into the egg or a lot of water left the egg. So our first egg was placed into just water. This is our control group. We just wanted to put it into plain water. Um, However, since we assumed that the water is definitely going to be a lot more diluted than the actual egg, the egg will have a lot more concentration within it. We knew that a lot of water would probably enter into the egg, so the egg would gain a lot of mass. So this was already known, but definitely we just wanted to do it with just plain water to see how that would go. Then our second egg was placed into 30 milliliters of salt, which is supposed to be our hopes for the isotonic solution, roughly probably what the egg also has within it. So no change in mass should be occurring with the 30 milliliters. That was just our hypothesis, which we'll get into after. And then 60 milliliters of salt, which is highly concentrated, and that means that water should be leaving the egg and going into the actual solution, since that's how osmosis wants to go. It wants to even it out. And then we did for egg 4 and 5 the same things, 30 milliliters and 60 milliliters, except with sugar. So our hypothesis, and this is the next step if you're doing an experiment, is to predict something that is going to happen from your project. So based on your background knowledge and what you know about the concept, what do you think is going to happen? And like I said, because of our general idea of osmosis and how it wants to basically even out the concentrations inside and outside of a material, the eggs that are placed in just water will expand. They'll take up more water since it already has such a high concentration within it. The eggs in water of about 30 milliliters of the solute we're estimating is going to be about the middle, so that will be the isotonic solution. It should stay about the same. And then the eggs in the water of about 60 milliliter of the solute should shrink. And so we collected our data by doing this. We had just 200 milliliters of water that the eggs were placed in, 200 milliliters of the water plus 30 milliliters of the solute, and then 200 milliliters of the water plus 60 milliliters of the solute. So those are our three different kinds, and we did it with salt and sugar. And we calculated the initial mass of the egg, the final mass of the egg, and then we calculated the percentage change to see how it changed. So in 200 milliliters of water, we could see how the mass of the egg increased as expected. This varied between the salt and the sugar. So for the salt, the mass did roughly the same. It was under a 1% decrease, so it was roughly the same. And for the sugar, it did increase by almost 2%, but it's still much significantly less than just having it in plain water. And then as expected, when it was placed into the 60 milliliter of the solute, the percentage change of the mass did decrease. So mass was lost from the egg, and this was expected. And then we were able to graph that data into these little charts, and you can see how for at least the salt, roughly around 30 milliliters is when there was no change in mass. So that was basically the isotonic solution. And then the change in egg mass when sugar was added, the isotonic solution was more so closer to 50 milliliters, roughly around there, which is different, but what we expected, somewhere between 0 to 60 is what we expected to be the isotonic solution. But to get an exact thing, we actually calculated how we can model this into a linear equation to model where that isotonic solution would be. And so we did exactly that, and we were able to see that it would be roughly between 31.4 and 31.5 milliliters, which is very close to our original hypothesis. This one is a little bit further off, but a little bit over 55 is where the isotonic solution would be achieving a 0% change in the mass when sugar was added. So yeah, just to talk about this project, we were able to prove that our hypothesis was correct, and the isotonic concentrations that we found for salt was 31.46 grams, and for sugar, 55.256 grams. In this experiment, one of the biggest things that was an issue was the validity. We kept a controlled environment, but we unfortunately could not do multiple trials because of limitations in our time. But definitely for the future, if you're doing your own experiment, that's something you should keep in mind. And for further experimentation, we would definitely want to test these calculated isotonic concentrations because now that is our new hypothesis. Our new hypothesis is that these are the isotonic concentrations, and it would be great to test that. And also testing other concentrations to see if we can expand our model on how osmosis is occurring in these eggs, get more reliable results because we're trying to make a linear prediction, but there's definitely not going to be perfect linearization of that. So having more data could definitely help with modeling that. And so basically, hopefully what you can gather from this video is that there are definitely a lot of small experiments that are easy to do. And this is one example of them if you want to model osmosis because you have an interest in biology, you're taking your biology class, or you're just interested in doing a little experiment on your own. But even if you weren't even if you weren't specifically interested in doing this experiment, it does show a pretty basic model for how you can do an experiment on your own, regardless of what topic it might be about. And this is just the general guideline of identifying your IV and your DV, developing a hypothesis and your plan, gathering your data, making it into graphs, analyzing it, and then drawing some conclusions and discussing the validity, what you can do further, what can you gather from this, and just reflecting on the overall experience. And so hopefully you're able to gather what a basic experiment would be if you're new to that. And so yeah, that's basically all that I have for you today. And hopefully I'll see you back in another video. Thank you. Bye.